Welcome to the Deep Dive, the show that gives you a shortcut to being truly well informed. Today, we're diving into what you might call the edge of life, that invisible yet incredibly dynamic boundary separating every living cell from its surroundings, the plasma membrane. Think of it not just as some static wall, but more like the cell's discerning gatekeepers, constantly deciding what enters and what leaves. This remarkable ability to discriminate is, well, it's absolutely fundamental to life itself. So today, we're embarking on a deep dive into the intricate, bustling world of cellular membranes. We're drawing our insights from a truly rich source, a core chapter from Campbell Biology in Focus.
Exactly. And our mission today is really to unravel the mysteries of this membrane structure. We want to understand how cells precisely control what crosses their boundaries and discover the sophisticated ways they communicate with each other. It's all about getting to the core of how life, at its most fundamental level, constantly interacts with its environment and coordinates these incredibly complex processes.
Okay, so to set the stage, let's look at something we all experience, learning. Our brain cells are constantly communicating. Tiny chemical signals, neurotransmitters, right? They get released from one cell and bind to specific receptor proteins on another. And this binding, it triggers the proteins to change shape, opening tiny passageways and allowing ions to flow in or out.
It's that continuous microscopic conversation, this chemical and electrical chatter across membranes that makes something as profound as learning possible.
So this deep dive, it should really illuminate the biological mechanisms behind such everyday wonders. So how does the cellular skin manage all this? Let's unpack the foundational concept. The fluid mosaic model. When you think of a cell membrane, don't imagine a rigid, static wall. Instead, picture something dynamic, ever-shifting. A vibrant collage.
That's a great way to put it. And the main ingredients in this dynamic collage are phospholipids and proteins. Let's maybe start with phospholipids. These are fascinating molecules because they're amphipathic.
Amphipathic, meaning?
Meaning they have both a water-loving or hydrophilic head and a water-fearing hydrophobic tail. Because of this dual nature, when you put them in water, they just spontaneously arrange themselves into a stable bilayer. The tails hide from the water on the inside, and the heads face outward towards the water. This self-assembly forms the very fabric of the membrane.
Okay, self-assembly. That's neat.
And what's really remarkable is that this membrane isn't static, it's fluid. Imagine a crowded room where the partygoers, the lipids and some proteins, are constantly shifting sideways. They're held together by weaker interactions, these hydrophobic interactions mostly, not rigid covalent bonds, so there's continuous movement.
So they just drift around.
Well, phospholipids move extremely rapidly laterally. Some proteins might drift more slowly, or they might even be anchored in place by the cell's internal skeleton, the cytoskeleton. It varies.
That fluidity sounds essential. But what stops it from becoming, too fluid or too rigid, especially if the temperature changes?
Ah, that's a critical point.
Yeah.
The membrane's fluidity is precisely regulated. It has to be just right. For instance, the types of hydrocarbon tails on the phospholipids play a big role. Unsaturated tails have kinks in them because of double bonds. These kinks prevent them from packing tightly together,
Okay.
which keeps the membrane more fluid, especially at lower temperatures. Saturated tails, on the other hand, are straight. They pack together closely, making the membrane more viscous or less fluid.
So kinks mean more fluid, straight means less fluid. Got it.
Right. And then there's cholesterol. At least in animal cells, cholesterol acts as a kind of fluidity buffer. At warm temperatures, like our body temperature, 37 Celsius, it actually reduces fluidity by restraining phospholipid movement. It kind of gets in the way.
Oh, interesting. So it stops it getting too fluid.
Exactly. But at cooler temperatures, it does the opposite. It prevents the membrane from solidifying by disrupting the tight packing of those phospholipids.
So it works both ways. Keeps it in the sweet spot.
Precisely. It's a remarkable adaptation to maintain optimal function across a range of temperatures.
And this is where we see evolution really doing its thing, right? Organisms in extreme environments actually re-engineer their cell membranes. Like fish in icy waters have more of those unsaturated kinky tails to keep their membranes flexible.
That's a perfect example.
Even winter wheat, apparently, adjusts its membrane composition in the autumn, increasing unsaturated phospholipids to prepare for freezing temperatures.
It's natural selection operating right there at the molecular level, a testament to life's adaptability.
Incredible. Okay, so phospholipids are the fabric. What about the proteins you mentioned, the mosaic part?
Right. If phospholipids form the flexible fabric, then the intricate designs and functional machines embedded within it are the membrane proteins. These proteins are the true workhorses. They determine most of the membrane's specific functions.
And what kinds are there?
Well, broadly, they fall into two main types. Integral proteins, which penetrate or even span the entire lipid bilayer, and peripheral proteins, which are just loosely bound to the surface, either on the inside or the outside.
And what do they do?
Oh, a huge range of things. Their roles are incredibly diverse. Some are transporters, acting as channels or shuttles for specific molecules. Others provide attachment points for the cytoskeleton inside or the extracellular matrix outside, helping maintain cell shape and stability. Many are crucial for cell recognition, identifying neighbor cells, and others are involved in signal transduction, relaying messages from the outside world into the cell. Some even act as enzymes.
Wow, okay. A lot of different jobs for these proteins.
Definitely. A functional mosaic.
And we can't forget the carbohydrates, right? You mentioned cell recognition.
Good point. Yes. The short-branched sugar chains, often attached to lipids making glycolipids or to proteins making glycoproteins, they typically face the outside of the cell and act like cellular ID badges. They are essential for cells to recognize each other.
Like name tags.
Kind of, yeah. A striking example is the human blood types A, B, and O. Those differences are determined by variations in the carbohydrate parts of glycoproteins on the surface of your red blood cells. It's a fundamental cellular fingerprint.
Fascinating. Okay, so we've got this fluid mosaic structure with lipids, proteins, and carbs. Now let's get to its most crucial function, selective permeability, the gatekeeper role.
Indeed. This is perhaps the membrane's defining characteristic. Its permeability varies greatly depending on the substance. Small, nonpolar molecules, things like oxygen, carbon dioxide, hydrocarbons, they're hydrophobic, just like the membrane's interior.
So they can just slip right through?
Pretty much, yeah. They dissolve directly into the lipid bilayer and cross easily, down their concentration gradient usually.
But what about the stuff that likes water? Hydrophilic things.
Ah, yes. For hydrophilic substances like ions, sodium, potassium, etc., larger polar molecules like glucose or even water itself crossing that hydrophobic core is a huge challenge. They can't just dissolve in it.
So they need help.
They absolutely need help. This is where transport proteins become absolutely crucial. They span the membrane and provide a pathway.
What kinds of transport proteins?
There are two main types. Channel proteins act like hydrophilic tunnels through the membrane. A fantastic example is aquaporins. These are specialized channels just for water. They allow billions, literally billions of water molecules per second to pass through, single file. It vastly speeds up water movement compared to diffusing across the lipid part alone.
Billions per second. Wow.
It's astonishingly efficient. Then the other type is carrier proteins. These are more like molecular shuttles. They bind to their specific passenger molecule, then change shape in a way that shuttles the passenger across the membrane.
So they physically move it.
Yes. For instance, the glucose carrier protein found in red blood cells binds to glucose, changes shape, transports it inside, and then reverts. It transports glucose something like 50,000 times faster than glucose could diffuse on its own. And it's incredibly specific. It won't transport fructose, even though it's a very similar sugar.
Specificity is key then. So the membrane's selective permeability isn't just the lipid barrier itself. It's this dynamic, elegant interplay between the lipids and these highly specialized protein gatekeepers.
Perfectly summarized.
All right, let's talk about traffic across the membrane. Sometimes substances move without the cell needing to spend any energy. That's passive transport, right?
Correct. Passive transport relies on the inherent tendency of substances to spread out. This is driven by diffusion. Diffusion is simply the spontaneous movement of particles of any substance so that they spread out into the available space. They move from where they are more concentrated to where they're less concentrated.
Downhill, essentially.
Exactly, down their concentration gradient. Think of dropping dye into water, it just spreads out naturally. That spreading out represents diffusion. It leads towards a dynamic equilibrium where molecules are still moving, but there's no net change in concentration. The concentration gradient itself provides the potential energy. No cellular energy, like ATP, is needed.
And a really crucial example of this for living systems is osmosis, which is specifically about water.
Yes, osmosis is the diffusion of free water across a selectively permeable membrane. Water also moves down its concentration gradient.
Okay, free water, what does that mean?
It means water molecules that aren't bound up interacting with solute molecules. If you have a high concentration of solutes like sugar or salt, there's less free water available to move. So water will move from an area of higher free water concentration, lower solute, to an area of lower free water concentration, higher solute.
Ah, okay. That makes sense.
And this leads us to the concept of tonicity. Tonicity describes how a surrounding solution will affect a cell's water balance based on solute concentrations.
Right, I remember these terms, isotonic, hypertonic, hypotonic.
Exactly. If a cell is placed in an isoponic solution, the solute concentration is the same inside and out. There's no net movement of water, and animal cells are stable in this state. Like our blood plasma is roughly isotonic to our cells.
Okay. What about hypertonic?
If an animal cell is in a hypertonic solution, meaning the solution has a higher solute concentration, then the cell water will rush out of the cell by osmosis.
In the cell.
Shrivels up. Think of microorganisms in a very salty lake, for instance.
Okay. And hypotonic?
The opposite. In a hypotonic solution, the solute concentration outside is lower than inside the cell. So water rushes into the cell. An animal cell in this situation will swell up and can actually cyclize, burst like an overfilled water balloon because it has no rigid wall to contain the pressure.
Right. That explains why if you put red blood cells in pure water, they pop.
Precisely.
But plant cells are different, aren't they? They have that cell wall.
They do. So in a hypotonic solution, which is actually the preferred state for most plants, water rushes in, but the cell wall prevents bursting. Instead, the plasma membrane pushes firmly against the wall, creating turgor pressure. This makes the plant cell turgid, or firm.
Which is why limp celery crisps up when you put it in water. It's taking up water and becoming turgid.
That's it, exactly. A simple, everyday example of osmosis and turgor pressure in action. In a hypertonic solution, though, plant cells lose water, and the membrane pulls away from the wall. That's called plasmolysis, and it causes wilting.
Got it. Okay. Still under passive transport, there's also facilitated diffusion. What's that?
Facilitated diffusion is still passive, meaning no energy is directly invested by the cell. Movement is still down the concentration gradient. But it uses those transport proteins we discussed, either channel proteins or carrier proteins, to help or facilitate the movement of hydrophilic substances across the membrane.
So it speeds things up for molecules that can't easily cross the lipid part.
Exactly. It speeds up their diffusion. Think of ion channels, for example. Many are gated. They open or close in response to a stimulus, like an electrical signal in a nerve cell, allowing potassium ions to quickly flow out. That rapid flow is facilitated diffusion through a channel.
Okay, so passive transport is all about moving downhill, concentration-wise, sometimes with help from proteins. But what if a cell needs to move something against its concentration gradient? Push something uphill from low concentration to high. That sounds like it needs energy.
It absolutely does. That takes work, and that requires the cell to expend energy. This is active transport.
Okay, tell me about that.
Active transport uses energy, usually in the form of ATP adenosine triphosphate, the cell's main energy currency, to pump solutes across the membrane against their concentration gradients. Critically, these transport systems are always carrier proteins, not channels, because they need to actively bind, change shape, and push the substance across against its natural tendency to diffuse the other way.
So they're like specific pumps.
Exactly. Active transport is crucial for maintaining internal concentrations of solutes that might be very different from the cell's surroundings, like keeping lots of potassium inside and lots of sodium outside a nerve cell.
Ah, the sodium-potassium pump. That's a classic example, right?
It's a prime example in animal cells. It's a molecular machine that oscillates between two shapes. It uses the energy from ATP hydrolysis to pump 3 sodium ions out of the cell for every 2 potassium ions it pumps in.
Against both their concentration gradients.
Correct. This maintains those crucial concentration differences vital for nerve impulse transmission, muscle contraction, and lots of other processes. Plus, by pumping out more positive charge than it brings in, this pump generates a voltage across the membrane. We call this the membrane potential. The inside of the cell is typically negative relative to the outside.
So it's like a tiny battery.
In a way, yes. This voltage, combined with the concentration gradient for an ion, forms an electrochemical gradient. This gradient represents stored energy that the cell can use for other work. Pumps, like the sodium-potassium pump that generate voltage, are called electrogenic pumps. Plants, fungi, and bacteria have a different main one, a proton pump, which pumps hydrogen ions out.
Stored energy, which leads us to another ingenious mechanism, co-transport. This sounds like getting a free ride.
Sort of, but it's indirectly powered by active transport. Co-transport happens when the downhill diffusion of one solute, moving along its electrochemical gradient, is coupled to the uphill transport of a second substance against its gradient.
Okay, how does that work?
Imagine the cell has used active transport, like a proton pump, to pump hydrogen ions, H+ out, creating a steep gradient. Now, those H+ ions want to diffuse back in, A co-transporter protein can harness that downhill flow of H+ back into the cell to simultaneously bring another molecule, like sucrose, into the cell, even if the sucrose concentration is already higher inside.
So the energy stored in the H plus gradient is used to pull the sucrose in.
Exactly. It's like water that's been pumped uphill. Active transport, creating the H+ gradient. Doing work as it flows back down, H+ diffusion. Carrying something else, sucrose, along with it uphill.
That's clever. And you mentioned this has real-world impact.
Immense impact. Understanding co-transport, specifically sodium-glucose co-transport in the intestines, led to life-saving treatments for severe diarrhea, especially from diseases like cholera. Patients are given an oral rehydration solution containing both salt, sodium, and glucose. The intestinal cells have co-transporters that take up sodium and glucose together from the gut.
Ah, so taking up solutes pulls water back into the body too, by osmosis.
Precisely. This simple biologically based treatment leverages the cell's own transport mechanisms to rehydrate the body and has dramatically lowered infant mortality from diarrheal diseases worldwide. A powerful example of basic biology saving lives.
Truly powerful. Okay, we've covered small molecules and ions crossing the membrane, but what about really big stuff? Large molecules, bits of food, even whole bacteria.
Good question. For that kind of cargo, the cell isn't limited to proteins embedded in the membrane. It uses specialized bulk transport mechanisms. These processes, exocytosis and endocytosis, involve packaging substances in membrane-bound sacs called vesicles. And importantly, they both require energy, usually from ATP.
Okay, exocytosis first. Exo sounds like exit.
That's right. Exocytosis is how the cell exports bulky materials. A vesicle, often budding off from the Golgi apparatus, moves to the plasma membrane, fuses with it, and spills its contents outside the cell.
What kind of contents?
Could be anything. Pancreatic cells secreting insulin, nerve cells releasing neurotransmitters into a synapse, plant cells delivering proteins and carbohydrates to build the cell wall. Lots of secretory processes use exocytosis.
Got it. So endocytosis must be the opposite, entering.
Exactly. Endocytosis is how the cell takes in material by forming new vesicles from the plasma membrane. The membrane sinks inwards, forms a pocket, pinches off, and brings the material inside in a vesicle.
Are there different types of endocytosis?
Yes, there are three main types. Phagocytosis, which literally means cellular eating. The cell extends projections called pseudopods around a large particle, like a bacterium or food debris, engulfs it and packages it into a large vesicle called a food vacuole. Amoebas feed this way, and our own white blood cells use it to destroy pathogens.
Okay, eating. What else?
Then there's penocytosis, or cellular drinking. The cell continually gulps droplets of extracellular fluid into tiny vesicles. It's not specific, it just takes in whatever solutes are dissolved in the fluid. It's a way to sample the environment.
And the third type. You mentioned specificity earlier.
Right, the third type is receptor-mediated endocytosis, and this one is highly specific. The outer surface of the membrane has special receptor proteins embedded in regions called coated pits. These receptors bind only to specific external molecules called ligands. Once enough ligands bind, the coated pit forms a vesicle, bringing the bound molecules specifically into the cell.
That sounds very precise. Is there a health connection here?
A critical one. Human cells use receptor-mediated endocytosis to take in cholesterol for membrane synthesis and other uses. Cholesterol travels in the blood in particles called low-density lipoproteins, or LDLs.
The bad cholesterol.
Well, it's necessary, but high levels are problematic. Normally, cells take up LDLs via specific LDL receptors using this endocytosis mechanism. But in an inherited disease called familial hypercholesterolemia, these LDL receptor proteins are defective or missing.
So the cells can't take up LDLs.
Correct. As a result, LDLs accumulate to very high levels in the blood, leading to plaque buildup in arteries, atherosclerosis, and a high risk of heart attacks, often at a young age. It really highlights how vital this precise transport mechanism is for health. And it's worth noting, these processes of exocytosis and endocytosis are constantly happening, adding and removing bits of membrane, ensuring the plasma membrane of a non-growing cell stays roughly the same size while also being continuously rejuvenated and remodeled.
Wow. Okay, so the membrane is constantly changing, moving, transporting. It's incredibly active. Finally, let's shift focus slightly to how the plasma membrane is absolutely central to cellular communication. This is the language that allows trillions of cells in an organism, like us, to coordinate everything they do.
Absolutely. Communication is essential. Cells communicate over various distances, sometimes as direct contact. Cells might have physical connections like gap junctions in animals or plasmodesmata in plants that allow signaling molecules to pass directly between adjacent cells. Or they might recognize each other through direct binding of surface molecules that's crucial in embryonic development and the immune response.
Okay, direct contact. What about signals that travel a bit further?
Then you have local signaling. Cells can secrete messenger molecules, called local regulators, that travel only short distances and influence cells in the vicinity. Growth factors often work this way. That's called paracrine signaling.
And nerve cells.
A specialized type of local signaling happens in the nervous system, synaptic signaling. A nerve cell releases neurotransmitter molecules into a very narrow gap, the synapse, separating it from a target cell, another nerve cell, or a muscle or gland cell. The neurotransmitter diffuses across the synapse and stimulates the target cell.
Connecting back to that learning example we started with.
Exactly. And finally, for communication across the whole body, there's long-distance signaling. This typically involves hormones. In animals, specialized endocrine cells secrete hormones into the circulatory system, which then travel throughout the body, but only affect target cells that have the specific receptors for that hormone.
Hormones can be very different types of molecules, right?
Oh, incredibly varied. From the simple gas ethylene, which promotes fruit ripening in plants, to complex protein hormones like insulin, which regulates blood sugar in humans. The chemical nature varies, but the principle of traveling far to specific targets is the same.
So how do cells actually receive, process, and respond to all these chemical signals, whether local or long distance?
That's the core of cell signaling. Thanks to some pioneering work, particularly by Earl W. Sutherland, studying how the hormone epinephrine adrenaline triggers glycogen breakdown, we generally understand this process in three distinct stages.
3 stages. Okay, what's the first one?
First is reception. This is the target cell's detection of the signaling molecule coming from outside the cell. The signaling molecule, also called a ligand, binds specifically to a receptor protein, which is often located on the cell's surface in the plasma membrane. This binding is very specific, like a key fitting into a lock, or maybe better, like a hand fitting into a glove.
And the binding causes a change.
Yes, the binding causes the receptor protein to change its shape. This shape change is the initial transduction of the signal. Crucially, only cells that possess the specific receptor protein for that ligand will hear the signal and respond.
So reception is detecting the signal. What's next?
Stage 2 is transduction. This is where the signal gets relayed and converted into a form that can bring about a specific cellular response. Transduction is often a multi-step pathway, sometimes called a signal transduction pathway. It typically involves a sequence of changes in a series of different molecules, often proteins called relay molecules. Think of it like falling dominoes.
A cascade effect.
Exactly, a cascade. One receptor activates another protein, which activates another, and so on. This multi-step process has a huge advantage. Signal amplification.
Amplification.
Yes. At each step in the cascade, the number of activated molecules can be much greater than in the preceding step. So the binding of just a few signaling molecules to receptors can trigger a response involving hundreds of thousands of molecules inside the cell. It makes the cell very sensitive to signals.
Clever. Okay, reception, transduction. What's the final stage?
Stage 3 is the response. This is when the transduced signal finally triggers a specific cellular activity. The response can be almost any imaginable cellular activity catalysis by an enzyme, rearrangement of the cytoskeleton, activation of specific genes in the nucleus, causing the synthesis of new proteins. The pathway regulates some cellular function.
Okay, let's dive a bit deeper into those receptors from stage 1. Where are they usually found?
Many, especially for water-soluble signaling molecules that can't cross the membrane easily, are plasma membrane receptors. There are a few major types. One huge family is the G protein-coupled receptors, or GPCRs. These are ancient and incredibly widespread, involved in sensing vision, smell, taste, and mediating the effects of many hormones and neurotransmitters.
GPCRs sound important.
Massively important. They're targets for something like 40–60% of all modern medicines. They work by binding the ligand, changing shape, and then activating an intercellular helper molecule called a G protein, which then goes on to activate another enzyme or protein in the pathway. Diseases like cholera and pertussis involve bacterial toxins that actually interfere with G protein function, showing how critical they are.
Wow. What other membrane receptors are there?
Another major type is ligand-gated ion channels. These are receptors that literally are channels. When the signaling molecule ligand binds, the protein changes shape to open or close a gate, allowing specific ions like sodium or calcium to flow across the membrane.
Down their electrochemical gradient.
Yes. This flow of ions can directly trigger an electrical signal, which is crucial for the rapid signaling in the nervous system.
Okay. Are all receptors on the surface?
No, not all. Some signaling molecules are hydrophobic enough or small enough to pass right through the plasma membrane. Steroid hormones, like testosterone and estrogen or thyroid hormones, and even the gas nitric oxide, NO, are examples. These molecules bind to intercellular receptors located in the cytoplasm or nucleus.
And what happens then?
Once the hormone binds to the intracellular receptor, the activated receptor hormone complex can often act as a transcription factor. It moves into the nucleus, if it wasn't already there, and binds to specific genes, turning them on or off, thus controlling the production of certain proteins. This alters the cell's behavior over a longer term.
So surface receptors for water-soluble signals, intracellular for lipid-soluble ones? Makes sense. Now, back to transduction, that relay race. You mentioned cascades. How do these molecular switches actually work?
A very common mechanism in these cascades is protein phosphorylation and dephosphorylation. Many of the relay molecules in signal transduction pathways are protein kinases. These are enzymes that transfer a phosphate group from ATP to another protein, usually activating it.
So adding phosphate turns it on.
Often, yes. It changes the protein's shape and regulates its activity. Then, to turn the signal off and reset the system, other enzymes, called protein phosphatases, rapidly remove those phosphate groups, inactivating the protein kinases.
So it's a dynamic on-off switch controlled by phosphorylation.
Exactly. It's a widespread mechanism for regulating protein activity. About 2% of our own genes are thought to code for protein kinases. That's how important this is. They regulate thousands of different proteins in the cell, controlling things like cell division.
And if that goes wrong?
Abnormal activity of protein kinases is implicated in many diseases, including cancer development. Many cancer drugs are designed to inhibit specific kinases.
Right. Are there other ways signals get relayed besides protein cascades?
Yes. Another important component in some pathways is second messengers.
Second messengers. So the ligand is the first messenger.
Precisely. Second messengers are small, non-protein, water-soluble molecules or ions that can spread rapidly throughout the cell by diffusion. Because they're small and water-soluble, they can quickly broadcast the signal from the plasma membrane to various parts of the cytoplasm. Common examples include cyclic AMP, CMP, and calcium ions, Ca2+.
Cyclic AMP, I've heard of that.
It's a classic example. Epinephrine, for instance, doesn't enter the liver cell itself. It binds to a GPCR, which activates an enzyme that produces a KMP. CMP then activates a protein kinase, starting the cascade that leads to glycogen breakdown. The binding of epinephrine can cause a rapid 20-fold increase in CMP concentration, quickly amplifying the message throughout the cell.
Amazing amplification again. Okay, so after all this receiving and transducing, the cell finally responds. What determines the response?
Well, the ultimate outcome is the regulation of some cellular activity. As we said, this could be in the nucleus or the cytoplasm. A nuclear response often involves turning specific genes on or off, thereby regulating the synthesis of particular proteins. This usually affects the cell more slowly, as protein synthesis takes time. A cytoplasmic response might involve directly regulating the activity of proteins already present in the cytoplasm. For example, epinephrine's effect on glycogen breakdown involves activating existing enzymes in the cytoplasm, leading to a rapid release of glucose for energy.
So the response can be fast or slow, gene-related or protein activity-related.
Exactly. And different cell types can respond differently to the same signal because they have different collections of receptors, relay molecules, and proteins to carry out the response. The whole system is incredibly specific and fine-tuned.
What an incredible journey we've taken today. Really, from the basic structure of the cell's edge of life, all the way to its complex communication networks. We've seen how these plasma membranes aren't just passive barriers, are they? They're dynamic, fluid mosaics, absolutely teeming with specific proteins and carbohydrates. They act as intelligent gatekeepers, efficient transporters, and vital communicators.
Absolutely. I think you've gained a powerful insight now into how life truly operates at this fundamental cellular level, how cells maintain their internal environment, how they absorb what they need, expel waste, and coordinate their every move with these really intricate signaling pathways. It's truly an elegant dance of molecules constantly in motion, all orchestrated by these incredible membranes.
So what does this all mean for you, the listener? Why should you care about this microscopic work?
Well, this fundamental understanding of cell membranes and signaling is really the bedrock for so much of modern biology and medicine. Think about how diseases like cholera or cystic fibrosis involve malfunctions in membrane transport, or how pharmaceutical companies design drugs specifically to target membrane receptors, GPCRs, ion channels to treat countless illnesses, everything from pain and allergies to heart conditions and mental health disorders.
So this knowledge directly impacts human health and our understanding of life itself.
Immensely.
Our deep dive today really shows that even at this microscopic level, life is just astonishingly complex and beautifully organized. It's like a constant, silent symphony of molecular interactions happening inside us all the time. So maybe consider this as we wrap up. How might unraveling even more of these intricate cellular conversations lead to new breakthroughs, maybe in personalized medicine, tailoring treatments based on an individual's specific signaling pathways, or even in advanced bioengineering?
Great questions.
What other secrets are these tiny dynamic boundaries still holding on to? Something to think about.